Lambda phage gene regulation is a complex process that determines whether the phage will enter the lytic cycle, that is replicating and lysing in the host cell, or the lysogenic cycle, integrating into the host genome and replicating passively. This decision is primarily controlled by the two key important proteins, that are cl and cro, and their interaction with specific DNA site. So cl is the lambda repressor and cro is the protein. So the cl is the protein and it is important for the maintaining the lysogenic state, and it acts as a transcriptional repressor, primarily targeting the promoter of the lytic gene, that is PR and PL. And cro is this protein is a key of lytic cycle. It acts as a transcriptional repressor, primarily targeting the promoter of the CL gene, that is PRM, thus inhibiting the lysogen. So PR and PL, these are the promoter initial rightwards and leftwards transcription respectively, which drive the expression of earlier lytic gene. The PRM is the promoter and responsible for transcribing the CL gene, thus maintaining the lysogenic cycle. There are operators OR1, 2, 3, 4 and OL1, 2, 3. These are the operator and these are the DNA sequence where the CL and CRO protein binds, control the transcription of nearby gene. Then regulation mechanism, lysogenic pathway upon infection of both PR and PL are active and leading to the transcription of earlier lytic gene and the synthesis of CL and CRO protein. So the CL dimerized binding of the OR1 and OR2, effectively repressing the transcription for PR and PL and thus turning off the lytic cycle. Simultaneously, CL dimer bind to the ORL1, activating the transcription for PRM, further promoting CL production and reinforcing the lysogenic state. So lysogenic state is stable with the phage DNA integrated in the host genome and CL repressing the lytic gene expression. Then lytic pathway, in that under certain conditions there is a DNA damage and the level of CL decreases, leading to the decrease in the repressor of the PR and PL. So CRO bind to the OR1 and OR3, further repressing the PRM and inhibiting the CL production, shifting the balance toward the lytic cycle. The Q protein, an anti-terminator, is expressed from the rightward operon and it facilitates the transcription of later lytic gene, leading to the phage replication and lysis. Then there is anti-terminator. N and Q protein act as an anti-terminator, allowing RNA polymerase to read through the termination site, enabling the expression of entire operon of the gene, and cooperative binding of the CL and CRO protein. The CL and CRO protein exhibit cooperative binding to the DNA, meaning that the binding of one protein molecule enhances the binding of another and making the regulatory switch more effective and switch-like. So the lambda phage effectively acts as a switch between the lysogenic and lytic cycle. The balance between the CL and CRO protein level determine the promoter activity and protein interaction, dictating which pathway the phage will follow. So this decision is influenced by the environmental conditions within the host cell, and such as the presence of the DNA damage or protease level. Thanks for watching. Do like, share and subscribe.